Hi, my name is Yug Yadava, and I did my project on the relationship between vitamin D levels and brain volume in children with demyelinating diseases of the central nervous system. This is just a brief abstract about my project. One of the demyelinating diseases I decided to focus on was multiple sclerosis, or MS for short. This is an autoimmune disease that targets the central nervous system, specifically the myelin, a protein that insulates nerve fibers. Although MS is a rare disease, it's even rarer in children. About 3 to 5% of all MS patients diagnosed will have their first clinical symptoms before the age of 18. Another somewhat new demyelinating disease that is in the scientific community is MOG antiviral disease. MOG is short for myelin oligodendrocyte glycoprotein, which is coated on the myelin sheet. Antibodies to this protein are responsible for the demyelination of the optic nerve, but it can also affect the brain and spinal cord. Although MS and MOG disease sound similar to one another, they are two separate demyelinating diseases. Neurologists and immunologists have been looking for ways to find new treatments and hopefully a cure to demyelinating diseases. And one of these new fields is vitamin D. Vitamin D has been known to regulate the immune system, and several studies have shown that those with higher vitamin D levels will have less autoimmune responses. One of these studies relating vitamin D to demyelinating diseases is Dr. Alberto Ascherio's study done in 2014. His study found that out of the 465 MS patients, those with sufficient vitamin D level, defined as above 50 nanomos over liters, will have a higher brain volume, lower lesion volume, and lower relapse rates than those with adult MS and had insufficient vitamin D levels. However, the association between vitamin D and brain volume in pediatric demyelinating diseases has never been associated before. So the objective of this study was to determine if higher vitamin D levels being associated with the regulation of the immune system will indicate a higher brain volume in pediatric multiple sclerosis patients that tested positive for MOG disease, and those with a monophasic demyelination, or MOG negative for short. Figure 1 here just shows the parts of the body where MS can affect. Figure 2 showing the frequency of MSs in certain countries. Table 1 showing us the vitamin D levels defined by the NIH, and Table 2 here showing the types of foods that have high concentrations of vitamin D. For this study, 59 patients were recruited from the Canadian Pediatric Demyelinating Disease Study at the Children's Hospital of Philadelphia, 12 of which were diagnosed with pediatric multiple sclerosis, 21 with MOG disease, and 26 with monophasic demyelination. Vitamin D, the month in which vitamin D was collected was also noted, as lower vitamin D levels are observed in the winter than they are in the summer. Brain volume was recorded in Z-scores. If a patient were to observe a negative Z-score, that means their brain volume is lower compared to their healthy mainstream peers. Crucial-Wallis tests were used as there was no normal distribution within the data, and linear regression tests were used to see if the association with higher vitamin D to higher brain volume was statistically significant. Age and gender ratios adhere to other studies conducted on pediatric demyelinating disease studies. With vitamin D, the lowest median value was in the MS group, with a median value of 43.65 nanomoles over liter, with the MOG negative group observing the highest vitamin D median value with 58.6 nanomoles over liters. The MS and MOG positive groups had a normal distribution for the month of collection. However, the MOG negative population in the MOG negative population, 17 out of the 26 patients had their vitamin D levels collected during the winter months, which were defined between October through March. Brain Z-scores of all three groups were in the had negative median values, and running Crucial-Wallis tests found no significant correlation among the three groups when it came to vitamin D, brain volume at onset, and brain volume at the 1-year follow-up. Figure 5 here shows us the gender of each of the groups. Figure 6 here shows the distribution of ages. Figure 7 here shows the interquartile range of the ages. Figure 8 shows us the vitamin D level distribution. Figure 9 shows the interquartile range of the vitamin D levels at onset. Figures 10 through 12 shows us the month in which vitamin D levels were collected among the three groups. Figures 13 and 14 gives us the distribution of brain volume at onset and at the 1-year follow-up. Figure 15 and 16 gives us the interquartile range of the brain z-scores at onset and at the 1-year follow-up. When running linear regression tests, it was found in all six that higher brain volume to higher vitamin D was not statistically significant when either at onset or at the 1-year follow-up. Figures 17 to 19 shows us the comparison of onset vitamin D levels to onset brain z-scores. And figures 20 to 22 shows us the onset vitamin D levels compared to the 1-year follow-up brain volumes. When it comes to vitamin D, it is important to note the month in which it was collected. Noting that there was a skewness towards the winter month in the MOG-negative group, the 58.6 nanomoles over liters could have been the same or even higher if there was a normal distribution observed, meaning that there is more compelling evidence that those with pediatric multiple sclerosis patients observe a lower vitamin D level, which could be specifically unique to this demyelinating disease compared to others. When observing brain z-scores, the insignificant result was noted as many demyelinating diseases tend to observe a negative brain z-score. When learning the linear regression test, there is some conflicting evidence as to why these came out insignificant. In Dr. Maury's study done in 2018, vitamin D levels compared to brain volume were not statistically significant. However, brain volume to a person's BMI was. However, it is important to note that vitamin D levels compared to a brain volume of a patient with MOG disease has never been done before. And with that, the University of Buffalo study from 2010 found that those with relapsing remitting multiple sclerosis and had insufficient vitamin D levels observed more brain atrophy than those with MS and had sufficient vitamin D levels. One major limitation of this study was the number of patients recruited. There were 59 pediatric demyelinating disease patients, while Dr. Alberto Ascherio's study had around 465 patients. In the future, more patients can assist with hopefully a significant evidence between vitamin D and brain volume, as noting in figure 22 and other figures from 17 to 22, that there was weak but positive trend lines observed. With that, looking now at lesion volume can also help give us a more comprehensive view of specific demyelinating diseases and how vitamin D can predict that. And finally, a more comprehensive disease course can give us a more further analysis of how onset vitamin D levels can affect a patient's course of disease over such an example 5 years. So to conclude, vitamin D levels were lower in MS patients than they were in MOG positive and in the MOG negative groups. There was no significant correlation between brain volume among the three groups at onset or at the 1-year follow-up, and 6 linear regression tests revealed that higher brain volumes to higher vitamin D levels were not significantly correlated to one another. In the following days, more patients and follow-ups along with investigating lesion volume to onset vitamin D levels will hopefully yield precise conclusions. A big thank you to the Children's Hospital of Philadelphia Department of Neurology for allowing me access to analyze part of the Canadian Pediatric Demyelinating Disease Study, and for allowing me to have the privilege of talking to advisors and doctors and about critiquing my project and how to make it better. These are some of the key references I used in this study, and this is an appendix. This is a table from Dr. Maury's study done in 2018. This is from Dr. Alberto Ascherio's study comparing brain volume to vitamin D levels, and figure 24 showing the frequency of MS types. And this is an example of Python code I used to derive histograms from. Thank you for listening, and I hope you found this study very interesting. And hopefully, with many more studies to come, and and many more updates on vitamin D levels compared to brain volume in pediatric dermatologic diseases, I hope new therapies and a hopeful cure will be soon to come.